Greetings. We're going to be talking about one of my favorite things, the BG, basal ganglia. This is just a review of where everything sits anatomically. The basal ganglia, located in blue, thalamus, purple, hippocampus, pinkish, amygdala is green, and the hypothalamus is right there, like hypo underneath the thalamus, right? And it's no coincidence that the basal ganglia is so close to all of these things. A lot of these things make up a ring called the limbic system. And that's kind of remembered by the mnemonic. It's like the 4 F's, like feeding, fleeing, something else, and effing. And you'll see sort of why. So the basal ganglia are gray matter. So they are ganglia, okay? They're neuronal cell bodies. Gray matter is cell bodies. That's like a super important thing to remember. Maybe I'll ask a test question on it. A gray versus white matter. And remember, in the brain, going from superficial to deep, it kind of starts gray, then white, and then gray again, because it's the deep gray matter of the brain. Whereas the spinal cord, remember, it had these like white matter tracks around the outside, and then this like butterfly looking thing that's gray matter on the inside. So it flips kind of, right? Whether you're talking about the brain or the spinal cord. Let's draw like a beautiful brain. Alright. The collections of nuclei that make up the basal ganglia include the caudate, which is probably the most well-known because it's huge. It's so big, they have the head, a body, and a tail. And they are also named separately because there can be different functions of the tail versus the body versus the head. I think one of them's involved in eye movement. Anyway, there's the putamen. There's the globus pallidus. The globus pallidus externa and interna. You'll also see the putamen and the globus pallidus together. They're called the lentiform nucleus. The caudate and the putamen together are called the striate. What's it called? Anyway, don't worry about it. Also not pictured that is part of the basal ganglia is the subthalamic nucleus and the substantia nigra. The substantia nigra can be divided into the substantia nigra pars compacta and the substantia nigra reticulata. And also, one of my favorite guys, the nucleus accumbens. And if you've heard of that, it's probably because of stories about addiction in the brain. The basal ganglia are very sensitive to extracellular dopamine. The nucleus accumbens is, some people say, is responsible for the development of addiction and addictive behaviors. And it's, strongly involved in the central reward pathway. Similar to the cerebellum, the basal ganglia is connected with everything, associated with a variety of functions. Motor movement is probably one of the biggest high yields. It's involved in voluntary motor movements, initiating and executing movement, and then inhibiting unwanted movements. Also in habit learning, right? That could be where some of the addiction comes in. Eye movements. I think it's caudate, I believe. Cognition and emotion. Initiation of movement and gross movement control. If you want to sort of sum up basal ganglia, Basal ganglia is very high yield because it's associated with a ton of diseases, and most of these diseases have something to do with a dysfunction in motor movement, which makes sense. Huntington's disease, right? They have something called chorea or choreiform movements. Huntington's disease is like terrible. It's like a combination of Parkinson's, Alzheimer's, and ALS. Like, it's awful. And you can get it really young, because this will probably be a board question for you, because of the concept of genetic anticipation. Because the longer it stays in the family line and you have more kids, the younger the kids will develop Huntington's disease. Parkinson's disease is due to a loss of dopaminergic neurons in the substantia nigra. Huntington's disease is the first thing you see is an atrophy of a caudate. So that's just implementing the different nuclei. Hemiballismus is where you have flinging of, like, say, like the left arm, and that's due to a lesion of the contralateral subthalamic nucleus. So if the left arm's flinging uncontrollably, the right subthalamic nucleus is compromised. Tourette's, all know what Tourette's is. So those are just some examples. Okay, so there's something called the direct pathway of the basal ganglia and the indirect pathway. And this just gives students hell. Like, students hate this. So I want to expose it to you a little bit. I kind of put this in as a joke. The cortico-basal ganglia thalamo-cortical loop. So it starts in the cortex, goes down, and comes back to the cortex. Okay, so direct pathway. Buzzwords. D1, because there's a dopamine 1 receptor and a dopamine 2 receptor. The direct pathway is associated with the dopamine 1 receptor. So the direct pathway, think about it as initiating and executing wanted movements, okay, wanted voluntary motor movements. The indirect pathway is associated with the dopamine 2 receptor, put R so you remember, and it's thought to inhibit unwanted movements. This is another way of putting it. The indirect pathway helps to prevent unwanted muscle contractions from competing with, I kind of like that explanation, voluntary movements. Dopamine is inhibitory on the indirect pathway via the D2 receptors. And so that's an example where the dopamine itself does not cause the action. It's the type of receptor that decides what's going to happen, because it's inhibitory when it's D2 receptors, but it's excitatory when it's D1 receptors. Okay. So what happens when you inhibit the indirect pathway? You get a net result of excitation, and then if you're excitatory, if you're stimulating the direct pathway, you also get the net result of excitatory action. So this is another complex example of disinhibition. There is something called the globus pallidus, I think it's externa, and that is normally kind of leashing the thalamus, preventing it from sending excitatory input to the cortex. So what happens as a result of we'll just take the direct pathway because it's the simplest example. The direct pathway results in inhibition of the GPE, therefore releasing the leash on the thalamus. Now it's free to send excitatory, I think a lot of times it might be glutamate, to the cortex. All right, and that is disinhibition because you're disinhibiting the GPE's effect on the thalamus. Okay, why is this important? Anytime you get the direct pathway compromised, you get hypokinesia, less movement than normal, lack of motion in our body. So I think, Parkinson's disease would be an example where the direct pathway is knocked out. And this is not like hard and fast rules, guys. There's a lot of overlap, but people like to try to boil things down in science, but it's not completely correct, but just a good learning tool. Anytime you interrupt or compromise the indirect pathway, generally, again, not always true, but you get hyperkinesia or dyskinesia, so too much movement or dysfunctional movement, which in general lead to production of additional involuntary muscle activity, like choreiform movements in Huntington's disease. Okay, circle this. What else? Like Tourette's would probably be an example of additional unwanted involuntary activity. Because remember what the indirect pathway does, inhibits unwanted movemts. Okay, so if the indirect pathway is compromised, you are no longer inhibiting unwanted movements, and you are therefore moving unwantedly. All right, let's do them. I'm just going to cover this real quick. Let's do glia. Glial cells make up, so we originally thought it was like a 10-to-1 ratio of glia to neurons. A lot more glial cells than neurons. Remember, glia are support cells. Now there's some studies that should just suggest it might be just a 1-to-1 ratio. I don't know, but regardless, glia are super important. Okay, in the CNS, you'll find 4 different types of glia: oligodendrocytes, astrocytes, ependymal cells, and microglia. Microglia clean up trash. They are specialized macrophages, so they take up debris, process it, and get rid of it so it doesn't build up in the brain. Ependymal cells make up the choroid plexus, remember, which makes CSF. Astrocytes do a bunch of stuff. They're really, really important. One thing they do is, I'll talk about it here. Okay. They have foot processes that during development they make contact with capillaries, and then they tell these endothelial cells to form tight junctions. And those tight junctions is what allows for a really, really selective blood-brain barrier. And the blood-brain barrier is made up of these endothelial cells with tight junctions and the choroid plexus epithelium. And remember, that's choroid plexus that's why bacteria and stuff like to enter. Oh, dude, nice, cool. Yeah, bacteria. Whoops. I can erase a lot easier. I'm like nerding out on this right now. Tight junctions. That might be a good tie-in, right? We talked about tight junctions. We didn't talk about it that much, but that is high yield, though, to know tight junctions of blood-brain barrier, and to know what makes up the blood-brain barrier, and to know the foot processes are involved in the integrity of it. Astrocytes are also involved in something called reactive gliosis. And that is whenever there's an injury in the CNS, these astrocytes have a compensatory response, and that's all you need to know. The astrocytes are responsive to injury. Okay. Oligodendrocytes are the myelin wrappers of the central nervous system. One thing about glia is, remember, for the most part, and we're now discovering this might not be true, but it's still taught that neurons are something called post-mitotic. And I don't know if you remember the cell cycle or not, but this is the I don't know, this is the stage of the cell cycle where they're off outside the cell cycle and they can no longer divide or regenerate. And, you know, there are definitely exceptions to this or not, but for the most part, they're post-mitotic. For that reason, glia are where tumors, like brain tumors, arise from. Like, I think the vast majority of brain tumors come from glia because they can undergo mitosis. Okay, and I think this will be a test question. It's really important because glia can divide, right? And you can only get cancer from cells that can divide. So neurons can't really give you cancer because they're post-mitotic. Glia can undergo mitosis, so you can get tumors. This is another reason that neurons are very, very, very sensitive to ischemic damage. I mean, we're talking about seconds, minutes of compromise of blood flow, and the neurons die. Okay, very sensitive. Something kind of interesting, there's something called CA1 neurons in the hippocampus, and those are responsible for memory. And they're one of the first to go in the hippocampus, so that's why people that get brain injuries and strokes, they lose memory pretty quickly and the ability to form new memories sometimes. Just giving you guys some high yield information. Also layers, I think 3, 5, and 6. There are like 6 layers of the histologically of the cerebral cortex, and these are also very sensitive to ischemic damage, like a lack of blood flow, lack of oxygen. So boards like to ask about these 2 areas because they're areas of susceptible neurons. Okay, in the peripheral nervous system, mainly 2 types, Schwann cells and satellite cells. Schwann cells are the myelin wrappers of the peripheral nervous system, and also of the areas of the peripheral cranial nerves, okay? And that might be weird to think about because the cranial nerves are doing their stuff up in the head face region, but they're like embryologically, they're kind of considered peripheral nerve systems. So you still have Schwann cells that wrap them, and there's a really famous tumor called a vestibular schwannoma, and that hits cranial nerve 8, and then for that reason you get unilateral hearing loss, and you get tinnitus, which is ringing in the ears. But yeah, that's basically a cancer of the cells that wrap the axons with myelin. Satellite cells, they often surround ganglia. This is my current my current project right now, actually. So there's this DRG, and there's tons of tiny satellite cells. And glia generally are smaller than neurons, but there's tons of these satellite cells that surround the DRG, and there's a lot of communication back and forth between the satellite cells. You'll also see the term like satellite glial cells. And they generally like surround neuronal cell bodies, and they're support cells, so they provide nutrition, metabolic support, all that stuff. Okay, let's stop here and then finish up next time.